Hello everyone, I'm going to do epigenetics. This is the third time I've recorded this because my PC just crashed and I just lost my videos and it was horrendous. Okay, so let's go start with epigenetics. What is epigenetics? Epigenetics is changes to gene expression without altering the DNA base sequence, without altering DNA base sequences. It comes in two forms, acetylation and methylation, but we'll get there when we get there. So how does it work? Well, it prevents transcription. In the exam, if it asks you what does it do, and I would not put protein synthesis, it prevents protein synthesis because it's a bit too vague. Go for transcription here. What should I? Why is it useful? Well, it's a response to an environment. This happens because it's a response. It's a response to environmental factors. So let's go to methylation. Methyl. Is on methylation? I think so. Methylation. So from GCSE, you know that methane looks like this: a carbon with a hydrogen and a hydrogen, another one and another one. Looks like this. And this is a gas. It's found in farts and cows and let's not go there. And the word meth, not as in the drug, is the same as meth here, but it also has a YL here. So a methyl group, where it's derived from, looks like this: a C, H, H, H. But there's no hydrogen here. There's an unknown bond. It's not bonded or anything. It wants to bond with something, but we haven't got there. We haven't got anything. Where does methylation occur? It happens at CPG sites. CPG. If you write it out in fully, it represents cytosine phosphate guanine. So it happens at cytosine phosphate guanine sites. That's where it happens. So if I draw out DNA, draw a deoxyribose, got phosphate here, another phosphate here, another one here. That's my deoxyribose here, and that's my phosphate there. And it's bonded to bases. For this example, I'm going to put a G here and a C here. I don't know if C can be on the top and G on the bottom. I don't know which way it's meant to be. Don't know anything about that. In Rich's videos, he did it like this, so I'm going to assume that this is correct because he's not generally wrong. On the other side of DNA, if I mirrored this, you would get your hydrogen bonds. I don't know if it's 2 or 3 here, and then your pentose. Upside down, isn't it? It's anti-parallel. It goes in the anti-parallel way. La 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 la. You know how that works. But in protein synthesis, in transcription, it's single-stranded, but it still forms hydrogen bonds. I think it's 3. I think it's 3 bonds. I can't remember, but I don't need to. But what else wants to form hydrogen bonds? Oh, guess what? We have a missing hydrogen bond here. So our methyl group, will come along with its hydrogens, and come along and go, ooh, hydrogen, and forms a bond here. If this happens, if a methyl group attaches here, it means RNA polymerase cannot bind. It cannot transcript. RNA polymerase cannot bind and therefore prevents transcription. Sorry, prevents though. Prevents transcription. Let's go to. Oh, here's a little bit of offspec actually. the methyl groups are added by an enzyme called methyl transferase. That is off spec, you don't need to know that, but something you write about in the essay if you wanted to. Or they could give you a question on, this enzyme is added to here. What if this enzyme is non-functional? What happens to epigenetics? Ooh. You could do that. I don't know. So, if I carry on, so let's go to acetylation. So, acetylation. Let's draw it out like we did with our methane. It looks like this. It's got a carbon, a double bonded oxygen, plus another carbon and 3 hydrogens, another carbon here, but nothing is attached here. There should be, but there isn't. And this is overall negatively charged. And shall I do? No, I'll write about it first. So where does acetylation occur? This bit is off spec. I'll do it in pink. Pink is off spec. That's not pink, that's black. Maybe pink. It happens at amino acid tails. And what you do need to know though, it happens at histones. So DNA is in the form of chromosomes, which is in the form of chromatin, which is supercoiled DNA, which in DNA is wrapped around histones. So if I draw some histones here, I'll save the histones for later, I need to draw them. Okay, histones, yeah. So if I draw some DNA, it's tightly bound around the histone proteins. And if I draw, how should I do this? Yeah, so DNA, let's draw some, if I draw DNA, and if I draw DNA like this in the fully form, and then here we have our sugar phosphate back there in the blue. So what is a phosphate group? A phosphate group is a PO4 minus 3 charge. So what happens if we add our acetyl with a negative charge to our phosphate, which is also negatively charged? They're going to repel each other because it's opposites attract, but negatives don't. It's like putting 2 magnets together. If you put 2 magnets, 2 north poles together or 2 south poles together, they're going to repel each other. So when you come along and put your acetyl groups here, what's going to happen is it's going to dewind. It's going to unravel the DNA slightly because it's negatively charged and it doesn't want to do that. So therefore, by unraveling it, our enzymes, this isn't RNA polymerase, can come along. It isn't RNA polymerase. Our enzymes can come along and transcription can occur. Transcription, yay! A little bit of off spec, a little bit more here. Like methylation, how methyl groups are added via methyl transferase, acetylation is added. Why won't my phone let there? It's added via the enzyme histone acetyl transferase, also known as HAT, which is, this is further, but this is not A-level, this is a bit higher than A-level biology, maybe degree level biology, and it's removed by the enzyme histone acetylase, which is HDAT. And that's acetylation and methylation done. Thank you. If you have any questions or if I've done any stupid mistakes, please tell me.